Hello, and thank you for joining our webinar, Imaging-Based Molecular Barcoding with Mid-IR Dielectric Metasurfaces. Before we begin, I would like to review some aspects of the webinar. We expect the webinar to take about 30 minutes. I would like to let our viewers know that they are welcome to ask questions at any time during the webinar using our Q&A window. We will answer all questions at the end of the event. If we not get to the questions during the webinar, we will answer the questions directly with you. I would now like to turn it over to Jeremy Roulette, our Senior Director of Life Sciences and Scientific, who will now introduce our guest speaker.
Thank you, Taylor. I'm honored to introduce our guest speaker this morning, Dr. Hatisha Altug, who's a professor at the Institute of Bioengineering at École Polytechnique Fédérale Lausanne, EPFL, in Switzerland. Dr. Altug received her PhD in applied physics from Stanford University, where I had the pleasure of meeting Hatisha over a decade ago. Her research today at EPFL focuses on nanophotonics for biosensing and has developed some novel surface enhanced spectroscopy techniques based on metasurfaces, plasmonics, and dielectrics. Professor Altug has received numerous awards, including the United States Presidential Early Career Award, as well as the prestigious Adolf Lohm Medal from the Optical Society of America, the European Research Council Consolidator Award, the ONR Young Investigator Award, and the NSF Career Award, among others. And it's, again, my great pleasure to introduce Dr. Altug, and with that, I'd like to hand it over to her for her presentation on imaging-based molecular barcoding with mid-IR dielectric metasurfaces.
Thank you, Jeremy, for the introduction. I also would like to thank for the invitation to speak in webinar. So welcome to all of you to my talk. So today I will be talking about on imaging-based molecular barcoding with mid-IR dielectric metasurfaces. In a broader scale, first in the introduction slides, I will describe how we use nanophotonics in my lab to introduce new generation of optical biosensors. So when we do biosensing, there are various questions that we would like to address. For example, if you have a sample that contains your analyte, so the first question is it there or not, or perhaps how much of molecule I have or this target analyte in my sample. What could be, for example, the composition of your sample? Maybe that could be a pathological sample. Or in the case of biological samples, for example, if it is a protein, what is the structure or conformation of the protein that you have? Or in the case of pharmaceutical applications, you may be wondering whether your drug candidate is interacting with the target protein that you are trying to address. And if so, what are the interaction kinetics? So a powerful optical technique that can actually address almost all of these questions is spectroscopy. So as most of you in the audience could be familiar, that the molecules consist of atoms that are arranged and connected in a certain way. For example, in the case of water molecule, we have hydrogen and two oxygens, and they are connected, then they will basically stretching, bending in a certain specific way. And the energy of these vibrational motions lie in the medium thread spectral range. So if you shine a broadband source, those vibrations will get absorbed at specific energies. So if you take out the absorption spectrum, you will actually see the fingerprint of a molecule that can pretty much be useful to answer the questions that I was just highlighting on the left side. So this is kind of highlighting the importance of the medium thread for chemical identification. So when we say about mid-IR, we are basically talking about, let's say, from 3 micron all the way to 12 micron range, and then you see that we can have the fingerprints of lipids, proteins, DNA, as well as some environmental targets that we may be looking for, sarin or explosives or some toxic gases. So it's a very useful technique because it's label-free, non-destructive. So it's like really for remote sensing of these reagents. And in terms of biology, I have been mentioning in the previous slides that we have the fingerprints of lipids, DNA, or proteins, which are the building blocks of, for example, cells. So we can see that with this chemical identification, we can look at pretty much complex samples. Not only we can identify the major biomolecules, but if you, for example, concentrate for the case of a protein, the mid-IR can even give an information about different molecular conformations. For example, beta sheet will have a different spectroscopic signature than alpha helix. So with mid-IR, we can actually resolve as well conformational motifs. While the technique is all very good, of course, it also comes with certain challenges. Some are more technical and some are more fundamental. For example, the sensitivity is pretty much fundamental. The signal scales in an exponentially to the absorption coefficient as well as the sample size, the thickness. So if you are talking about with monolayer samples or like monolayer thick lipid membranes, you can we are basically talking about samples on the order of 1 to 10 nanometer or so. So this is pretty small compared to the long wavelength of mid-IR photons. So that's why it's pretty much challenging to do like low analyte concentrations or monolayer studies. Another challenge is more for biology, let's say the water, which is where the biomolecule resides, have strong absorption bands that can obscure the signatures of the analyte samples. And another technical challenge is that the current state of the art heavily relies on Fourier transform infrared spectroscopy, and these are like massive instruments still at the moment. They are bulky and expensive. And this is where basically we see as our field, nanophotonics can address some of or perhaps all of these challenges. And the strength of nanophotonics is that we use nanoscale antennas or resonators that basically confine the light to nanoscale volume in such a way that the long mid-IR photons, the size of long mid-IR photons within this hotspots of these antennas can be comparable to the size of the molecule. So we are basically overcoming the size mismatch issue. And then we can furthermore enhance the light-matter interaction to achieve better sensitivity and improve the signal of the molecule compared to the background signature, for example, for water. And furthermore, as everything is operating in the nanoscale on the chip-based platforms, we also have chance to do on-chip integration. So in my group, we have been looking various nanoscale structures, and as well as in the field, some are actually using plasmonics. We also initiated one of the first works on graphene plasmons for tunable mid-IR biosensors. And most recently, we also got into the dielectrics, which I will highlight within the rest of these talks, that we can get high-Q resonators that can open up a really interesting imaging-based mid-IR biosensors. The state of the art actually has been mostly focusing on plasmonic structures. So, and this is kind of lying the initial fundamental work of so-called surface-enhanced infrared spectroscopy, or SEIRA, with engineered plasmonics. So these are top-down fabricated resonator structures. So with these ones, you can have pretty much high sensitivity. So this is one of the first works that we did in our group for detection of protein, and there are various other groups within, pretty much across the world working in this area. And the plasmonics is powerful. It's biocompatible. It has strong resonances, and you can look at complex samples, for example, lipid, protein, and their interaction, or you can even look at real-time in situ protein conformational changes. But the plasmons have certain limitations which we think we can address with dielectrics and come up with even new way of interrogating mid-IR absorption signatures. And to understand that, first, we need to see what we can achieve if we go from plasmonics to dielectrics, and that will actually be in the form of reducing the resonant line width. So what I mean by that is actually expressed in this slide. So the resonant line width or the sharpness is qualified quantified by so-called the quality factor of the resonator, and it's given by the resonant line width, resonant frequency divided by the line width. For the case of plasmonics, where we use metals, if you work in the mid-IR range, independent of whether you work with silver or gold or copper, compared to the visible spectrum, you will not see much changes, and they will be all working like a true metal. And the quality factor of the resonators, of these plasmonic resonators, will be less than 10. So they will be relatively broad. Let's say if we convert this within the range of 1600 wave numbers, so you will have line widths on the order of 200 inverse wave numbers. So what does it mean for biomolecular sensing? For the case of absorption lines of proteins, for instance, the amide1 and amide2 absorption bands line widths are on the order of 40 to 50 wave numbers. So you basically see a little dip in this broad resonance peak of plasmonic resonator. And that's basically the typical signature of a plasmonic-based sensor. And that's also what I have been just highlighting, for example, in this slide or in this slide in the literature. So the question that we started to ask a couple of years ago is that what happens if we have a high Q resonator? So now the high Q resonator is, of course, you need to compare with the resonant line width of the protein absorption line. So as I mentioned, that's on the order of 40 to 50 wave numbers. So now if I make a resonator that has a line width of 10 or even less wave numbers, so that will be actually sampling some of those absorption lines at a given specific wave number. So as these resonators are higher Q compared to, let's say, the plasmonics, so when I introduce a lossy material, in this case an absorbing protein layer, this will represent itself mostly as a strong absorption, a strong suppression in the reflection signal, as well as perhaps some of another effect could be broadening, but the majority will appear as a strong dip in the reflection. So the question is, so you have this spectrally selective detection at a specific wave number, how we can convert this into spectroscopy. And this is actually something that we kind of introduced in this recent paper that has been published this summer, several months ago. So as I mentioned, you have a high Q resonator, which we actually achieved through the use of this dielectric resonators. And then what we did is, in order to cover a wide spectral range, we made arrays of them. And this array, in this case, I show a representation of 5 by 5 dielectric metapixels. So this is a pixelated metasurface. So one of those, 25, is indicated by this orange line and has a specific resonant wave number corresponding in the bottom. So and then what you have is in another pixel of this metasurface indicated by this green box, you will have a different spectral frequency. So what I do is actually within this 25 different samples, I can, due to the high Q resonators, I am able to do a one-to-one mapping between spectral information and spatial identification. And this is something that we achieved with this specific design that has been outlined in this paper of this zigzag arrays of ellipses. And these structures actually gave high Q resonances, but as well as what we emphasize is spectrally clean resonances. So these are all these 25 metapixels correspond to the same unit cell, but a different scaling of it. Okay, so we basically scale it from 1 to 1.24, and then as we scale the resonances, you see that within this broad tuning range, the only one resonance show up, and then there is no like other resonance modes popping up, and that's what we mean spectrally clean resonator. So that was very important for this implementing to this concept. As well as another advantage of this design is that it gives strong field enhancements or strong near fields, but they are outside the dielectric, so they are highly accessible to the analytes that we are trying to probe with this metasurfaces. So this is another novelty that I should emphasize, so that the near fields are not confined within the resonator, but they are outside and accessible. To explore this one-to-one mapping for fingerprint retrieval, what we do is we take this pixelated metasurface and cover with an absorbing layer. In this case, in this simulation, we actually put a protein monolayer directly in contact with the metasurface. And this protein has, of course, specific fingerprint. For instance, it has MI1 and MI2 peaks at 1650 and around 1550 Way number. And of course, the pixels that are overlapping with the absorption peaks of this analyte, they will be suppressed. So in this case, the green is suppressed compared to all other pixels, and then the next one that is highly suppressed is the yellow one, and the pixels that are not overlapping in terms of absorption fingerprints of the analyte, in this case, let's say blue and green, they will basically will not get changed. So if you look at all these 25 pixels, you basically see that from the trace of the envelope of these reflection peaks, you can correlate exactly the absorption fingerprint. So the dips corresponds to the peak itself. So this is more like, let's say, the spectroscopic response. You can pretty much see that that will correlate itself into a two-dimensional imaging barcode. So the region, the spatial region indicated here with a box of 2 is overlapping with the highest absorption peak, and then you kind of see that shows up as the darkest pixels compared to the rest of the pixels. So this absorption fingerprint is basically converted into a two-dimensional molecular barcode. So pretty much we digitize this molecular fingerprint information and convert it into an image representation. And so to demonstrate this experimentally, actually we went and fabricated these structures. So they are made out of amorphous silicon layer that is deposited by PECVD on a magnesium fluoride substrate, magnesium fluoride IR transparent substrate. And we used EV lithography and dry etching techniques to fabricate these pixelated metasurfaces. In the experiment, we actually did 10 by 10, so we have 100 pixels, and each pixel is containing this zigzag arrays of dielectric resonators over an area of 100 by 100 micron, and the spacing between each pixel is 40 micron. And the scaling that I was mentioning to you is basically where we tune, where we scale up, so to say, the ellipse long and short axis A and B, the periodicity P and X in both directions. And we kept the height same, so it's from the same deposition. So the height is about 700 nanometer, and also we kept the tilt angle at 20 nanometer, and the rest is just scale, let's say like a PowerPoint, scale function that we just zoom in from 1 all the way to 1.34. And so to characterize this large area sample, that's actually what we use very conveniently, the spero microscope, because it's one of the unique systems that is giving a large area mid-IR imaging capability. So we use 4x IR objective, which gives an outstanding field of view of 2 by 2 millimeter. So this is actually perfectly matching to, is quite ideal to look at this such large area samples. And then this is, projected in the imaging detector of the built-in camera within the spero system. So you kind of see that by looking at different wavelengths from this tunable QCL sort that is again built in the spero. For example, when we shine and look at 1300 wave number, you basically see the pixels, the pixels at the low end of the metasurface is lighting up, and if we increase the tuning wave number all the way towards the 1800 wave number, so this is 1770, you basically see the pixels that are on the upper right hand side of the pixelated metasurface are lighting up. So we basically, show that, the metasurface is covering from around 1300 all the way to 1800. And, this one is, this one is just showing some selected pixels, but if we actually show the entire 100 metapixel, you kind of see that we very nicely sampled. The resolution that we choose is about 4 wave number to be comparable with state of the art FDR samples, and all these 100 pixels have almost uniform resonant line width and reflections, responses, and full width half maximum. So the quality factor of all of them are above 100. So it's pretty uniform. Now to show kind of the experimental implementation of the simulations that I was presenting in the beginning. So we have this 100 metapixels. So some of them are highlighted for the clarity. So when we absorb a protein, in this case we use a physioabsorption, so the protein is in contact with the metasurface, you basically see that the pixels corresponding to the or overlapping with the absorption peaks of this protein is diminished. So the envelope nicely traces out the absorption fingerprint of the protein. And this is kind of what we extract in these in the right-hand side. So the colorful one is extracted from our metasurface pixels. That's why they are actually given in this color coding. And then these 2 peaks are associated with amide1 and amide2. As a reference, we also showed what should have been the signature of the same protein if we obtain with IRS measurements coming at a grazing angle, where we multiply further 60 to be comparable with the signals that we get. And then you see that there is a very good agreement with this independent IRS measurement. So it's highly and nicely reproducing the spectrum where we extracted from this pixelated sensor. And what we are actually envisioning is that this type of system could be quite interesting for a spectrometerless operation. So our vision is that you can use a broadband source because each pixel will give you the spectroscopic information. So the spectroscopic filtering will be done by the metapixel. Furthermore, you will have the near-field enhancement by the resonators. And if you use a broadband detector that are, associated with each pixel, you can actually read out the response from each pixel in an imaging format. So this is what we call spectroscopic integration. So that's what the detector will be if you shine with a broadband source for each metapixel. So if there is an overlap with the absorption, it will be reduced in amplitude, so the integral under that area will be representative of that, that peak change. And in fact, if we implement this integrated absorbance approach for the data that we obtained from the SPERO system, we actually get this molecular barcode that is obtained within this so-called spectrometerless or with this broadband illumination scheme. And then this will be again the imaging-based molecular barcode in this integration. And we kind of, we show this in the reflection because that's what we operated within SPERO, but the system is also applicable to operate in the transmission. So you can imagine that you have a broadband source, you have your pixelated metasurface, which gives the spectral information as well as signal enhancement that is pretty much in contact or maybe in close proximity to your imaging detector, so you can kind of line them up and integrate them in a miniaturized manner. So that's kind of what we believe what we show that it can, in the simulations it can happen, and the next implementation will be to do this, of course, experimentally. And using this integrated absorption method, we actually showed that you can get chemical identification. So this was the molecular barcode that I was showing with a protein sample. We also put on these structures a thin layer of PMMA, which more represents the kind of the application for material science. You kind of see that this absorption fingerprints of PMMA is much different than protein. So this is converted in a molecular barcode, again, a completely different response. And the third example is for more of an environmental monitoring, where we took glyphosate, and again, you get a completely different imaging representation. So you can actually do at this stage, so you can do an image-based pattern matching to your target analyte, especially if you are interested in chemical identification. But you also have the freedom, of course, if you do, if you calibrate each pixel, you can convert these images, and you can extract the spectra, and then you can kind of continue with the spectroscopic methods. But we believe that there is quite a bit of advantage to stay in the image representation. So chemical identification is one step, and the next step could be is that once you identify your chemicals, you can also do compositional analysis. And to show this, we did a proof of principle experiment in this paper. We actually used a polymer mixture that contains a mix of PMMA and PE, where we kind of mixed ourselves in the lab setting, and then we had this predetermined mixtures. And we also know our, let's say, so-called the database. For example, if we have pure PMMA, we know how it should look like, or if we have pure PE, we also know how it should look like. And then when we have different, ratios of it, from this image, if we know this, the base, let's say, so-called X and, XP of, or XPMA, so we can actually extract different coefficients that we highlighted here in terms of beta. So, from the mixture we already know, but when we kind of read out from these images, and then we did the linear composition, we got a very good match between what we put and what we extracted with the image. So this is actually where we see as the next, let's say, area to explore. So you may kind of think that, I will have a mixture, and then it will look pretty like unobvious to you what this, mixture contains. So if you have some library, a barcode library that we know of, let's say you have 4 different target analytes that you may be suspecting, then you can do certain image processing techniques. For example, one could be a very simple linear decomposition, and then you can identify, what is the percentage of each analyte. But of course, there are other aspects that can be further implemented. You can, as opposed to simple linear decomposition, then you can think about more advanced image analysis techniques, especially if you have a very large database where you have a large barcode library, then you can do more advanced pattern recognition by some machine learning algorithms or artificial intelligence. So that's where we think this new sensor platform could benefit from. So I guess in conclusion, mid-IR is a very powerful spectral range, in particular for biosensing, because it's label-free, chemical-specific, and non-destructive. But current mid-IR spectroscopy instruments face limitation for the sensitivity, for the fact that they are bulky and expensive. They have limited integration capabilities, and that's where we think that large area imaging techniques is really powerful for spectroscopy. So for the case of dielectric metasurfaces, they are really interesting because they are CMOS compatible. They are on-chip technologies, so they can be integrated with large area detectors and light sources, and that they can be used to deliver miniaturized spectroscopy tools. And especially if you stay in the large area imaging platforms, you can also think of that this opens up the door to use data science tools where we can really do smart sensing approaches. So with that, I would like to acknowledge my lab members at EPFL who contributed this work, as well as my colleagues and collaborators, in particular for the dielectric metasurfaces from Australia, Yuri Kishvar and Dragomir Neshev's lab. And I also would like to acknowledge my funding sources from EU in terms of Consolidator Award and Marie Curie postdoctoral fellowship opportunities, as well as the clean room of EPFL, which was very instrumental for fabricating this large area pixelated metasurfaces. With that, I end my presentation, and I will be happy to take your questions.
Yeah, thank you, Dr. Altug. So as you mentioned, we have time for some questions at this point. But before we get into that, I want to say thank you for that excellent presentation. It's really groundbreaking research covering a number of disciplines from nanofabrication to microfluidics to monolayer spectroscopy and imaging. So thank you for that. Okay, so some questions are coming in. I think as they do come in, I have a couple to begin with. One of the questions is, how can you talk a little bit about how you get the analytes in close proximity to your dielectric sensor array?
Yeah, so thank you for this nice question. So it's first of all, I have to emphasize that this is a surface-enhanced technique, spectroscopy technique. That means the analyte needs to be really in close proximity with the metasurfaces. So the field extends within, let's say, up to about 100 nanometer or so. The analytes have to be in close proximity with the metasurfaces. For the applications that I showed demonstrated here, for example, for the proteins, they were directly, introduced on the meta chip, where they basically incubated. We weighed enough, and then we washed. So they are in physical contact and introduced by physioabsorption. For the case of PMMA, this polymer PE and PMMA, we did spin coating of a very thin layer, less than 10 nanometer thick. So you have to find ways, either by pipetting or maybe some microfluidics or spinning, that you have to introduce your analyte and bring it really in close proximity with the metasurfaces.
Yeah, thank you. So I guess as a follow-up, are those substrates reusable in principle?
Yeah, in principle, they are reusable. So these are silicon on calcium fluoride substrate. So this is pretty robust. So you can use some cleaning procedures. With there are various in literature from piranha cleaning to UV cleaning or oxygen plasma, depending on what your analyte is. If it's polymer versus like biomaterial. So in principle, you can actually implement as long as it doesn't degrade your metapixels or substrate. You can clean and you can reuse. But of course, if you this also kind of opens up another direction, as these are CMOS compatible structures, and the structures are also not very small. You can also think of that they can be fabricated with low-cost techniques. So at the moment we use e-beam lithography, but you can imagine that they can be fabricated with low-cost techniques. So you can also consider them to be disposable if that's what the application may require.
And you mentioned magnesium fluoride as one of your materials. What was it like working with that material? And is that, would you consider that CMOS compatible?
Yeah, so that's a good question. So we have been using magnesium fluoride, but now we also use calcium fluoride, and of course it's not like a let's say silicon wafer, but I think maybe CMOS friendly is more more appropriate than just CMOS compatible.
Okay. And then one of the questions that came in is, how do you determine the size of your dielectric resonator pixel?
Mm-hmm. Okay. The size of, so in this case, the size we choose as 100 micron by 100 micron. And so the size you would like to perhaps arrange, depending on what is your field of view, what will be the spectral resolution that you would like to have within that field of view, and what will be the accommodating structure to get enough signal. So if you reduce the size from 100 to lower, let's say 50 wave number, then of course you will reduce the signal from each pixel, but then you will able to pack more number of pixels within the given field of view, which means you will have a better spectral resolution. So at the end, it's the kind of the application based on your requirement. Perhaps you can compromise a bit from the resolution. In this case, we did 4-way number resolution just for clarification, so which is already state of the art, and it's like 100 micron, and the structure, is less than 2 millimeters. So this is, directly matching to Sparrow's like field of view. You can optimize further. In fact, we have been looking a bit smaller sizes, but you don't want to go way too small. So at least maybe the ultimate case will be where one metapixel is corresponding to one imaging detector pixel.
I see. Okay. And I think just to wrap up, running out of time, and I know there are a number of questions we'll probably answer offline, but just, in realizing your vision of kind of a portable type system, what do you think are really the next steps of your research to get to that goal?
Well, so I think we have a pretty good setting at the moment in terms of, fabricating the structures over large areas. So the next steps will be to really implement the integration. So bring up the parts, and for example, I mentioned a broadband source. Then we need to investigate, whether we have enough signal for integrating over such large areas. Putting a suitable detector, an imaging pixelated imaging detector, and what should be the specifications of that, in terms of how many pixels, what is the sensitivity? Do we need to cool it or not? So I think it's more of like the next challenge is really integrating and putting all the aspects of from finding the right elements and finding if the power, the sensitivity is or all these parameters are sufficient enough to integrate them. And then that will be more like a prototype demonstration, and then one can imagine then to take that prototype into a real device.
Okay, well, thank you so much for that presentation. I think that's all the time we have now. If we didn't get to your question, we'll certainly follow up with you after the webinar or put you in direct contact with Dr. Altug. So at this time, I'd like to thank Dr. Altug for her presentation and the time she spent with us today covering her research, which truly is, I think, very novel and groundbreaking in a number of aspects. So thank you, Hatisha, for your time today.
Thank you again, Dr. Altug.
Thank you, Taylor.
And thank you to our viewers for joining us today. If you enjoyed this webinar, please take a few moments to fill out a brief survey. Thank you again, and have a great day.
Bye all.